1898, a man named David de Rosier wrote an article in the journal Cell, which is a very prestigious scientific journal, entitled "The Turn of the Screw, the Bacterial Flagellar Motor." Now, David de Rosier is a professor of biology at Brandeis University in Massachusetts and has worked on the bacterial flagellar motor for most of his career. Now, in that article, he makes a statement, quote, "More so than other motors, the flagellum resembles a machine designed by a human," close quote. So David de Rosier also recognizes that the structure of the flagellum appears designed.
What I wrote was, this is a machine that looks like it was designed by a human, but that doesn't mean it was designed. That is the product of intelligent design. Indeed, this more has all the earmarks of something that arose by evolution.
Using an electron microscope, de Rosier produces ghostly pictures like this one, revealing the inner workings of what's been called the world's most efficient motor.
This is the drive shaft. This transmits this torque generated by the motor. That would then turn the propeller, which would push the bacterial cell through the fluid.
Michael Behe has argued that the flagellum could not have evolved, since its parts have no function for natural selection to act on until they are fully assembled. But evidence that refutes Behe's claim of irreducible complexity comes from a tiny syringe that injects poison, found in some of the nastiest of all bacteria.
This is a structure found, for example, in Yersinia pestis, the bacterium that causes the bubonic plague. And look at the similarities. Now, this structure doesn't rotate, but it still has to extend this structure, which is equivalent to the rod, the drive shaft here. It has to extend that because it needs this little channel. It's like sort of like a syringe. So the virulence factors that are made inside the cell, which is down here, can be exported, pushed up into this hole, and exported out through this long kind of needle, perhaps into a cell in your body or mine, and thereby create misery.
And it turns out the two structures look similar for a reason. The syringe on the right is made of a subset of the very same protein types found in the base of the flagellum on the left. Though the syringe is missing proteins found in the motor and therefore cannot produce rotary motion, it functions perfectly as an